There is something sitting in your kitchen right now that most people throw away without a second thought. Maybe it is the papery shell of a pea you shelled last week. Maybe it is the fibrous casing of a bean you cooked for dinner. Something you opened, used the inside, and discarded the outer structure automatically, the way you discard packaging. And that is completely normal. That is what almost everyone does. But here is what changes everything. Researchers in metabolic science and nutritional biochemistry have spent decades studying the exact structures that end up in your compost bin. Not the seeds inside, not the beans or legumes themselves, the pods, the casings, the outer shells, the parts that get discarded reflexively, as if their only purpose was to protect what was inside and then disappear. What they found changes how you look at every plant you have ever handled. Some of these structures contain bioactive compounds that interact with the way your body processes glucose. Compounds that slow the absorption of sugar into the bloodstream, influence how insulin responds, reduce inflammatory markers associated with metabolic dysfunction, and in some cases appear to activate cellular pathways that improve the body's sensitivity to its own insulin. Almost nobody knows this, not because the research is hidden. It is published, peer-reviewed, sitting in journals that most people will never read because there is no commercial reason to make this information widely accessible. If you are new to this channel, subscribe now, because what is coming in this video is the kind of information that genuinely changes how you think about food, metabolism, and what you might have been discarding without realizing its value. 12 seed pods, 12 structures that billions of people discard every single day, structures that traditional cultures across 6 continents used intentionally for centuries, and structures that modern researchers are now examining with tools those traditional cultures never had. The further we go in this list, the more remarkable the science gets. And the final entry has more accumulated human research behind it than almost anything else in this category. We start at number 12, the pigeon pea pod. If you have spent time in South Asian, Caribbean, or East African cooking, you know the pigeon pea itself, cultivated for over 3,500 years. But the pod, the outer shell holding those small round legumes, almost always goes directly into the discard pile. The pod wall of the pigeon pea contains flavonoids, specifically orientin and vitexin, which have been studied for their ability to inhibit alpha-glucosidase, an enzyme in the digestive tract that breaks complex carbohydrates into simple sugars for absorption. When that enzyme is slowed, glucose enters the bloodstream more gradually. The post-meal glucose spike is reduced. It is a modest effect at this level. Interesting, but modest. That is why it opens the list rather than closes it. The same basic mechanism, that slowing, that modulation of how quickly glucose becomes available, appears in every single pod on this list. What changes is the degree, the mechanism, and the complexity of the interaction. Number 11 is the honey locust pod. The honey locust tree, Gladitsia triacanthos, is so common in North America that most people have walked past it hundreds of times without a second thought. It produces large, flat, reddish-brown pods containing a sweet pulp surrounding the seeds. That sweetness is interesting on its own. What researchers found in the pulp and pod wall is a combination of galactomannins and polyphenols that work together more effectively than either compound alone. The galactomannins form a viscous gel in the digestive tract, physically slowing carbohydrate absorption. The polyphenols reduce oxidative stress markers associated with metabolic syndrome. Indigenous peoples across North America, the Cherokee, the Meskwaki, various tribes through the eastern woodlands, used honey locust pods in teas, preparations, and direct consumption for centuries. Then modernization happened. The honey locust became an ornamental tree or a nuisance species. Its pods became lawn debris. The knowledge of what those pods contained faded because there was no economic incentive to keep it alive. That pattern repeats across this entire list. Traditional use, forgotten knowledge, then rediscovery through laboratory analysis. It repeats so consistently that it stops feeling like coincidence. Number 10 brings us to the ordinary bean pod. The common bean, Phaseolus vulgaris, is one of the most widely consumed legumes on the planet. When those beans are prepared, the pod gets discarded almost universally, composted if someone is thoughtful about it, thrown away if they are not. The pod wall of the common bean contains phenolic compounds, including chlorogenic acid, quercetin, and kaempferol, at concentrations that are significantly higher than the bean itself in some varieties. Chlorogenic acid has been studied extensively for its ability to reduce intestinal glucose absorption and to modulate the liver's production of glucose through a pathway involving glucose 6-phosphatase. This is the same compound that makes coffee metabolically. Interesting. People have been unknowingly consuming chlorogenic acid in their coffee for centuries. The bean pod contains it in concentrated form, in the structure that gets thrown away before the cooking even starts. The part you discard may contain more of the compound than the part you eat, and this is only the beginning of the list. We have not yet reached the pods that shift the conversation from interesting to genuinely remarkable. Those are coming. And the gap between where we are now and where this list ends is wider than most people would expect. Pea pods are so ordinary that most people have stopped seeing them entirely. You buy fresh peas, you shell them, the pods go in the trash, or you buy them frozen, already shelled, and the pods never even enter your kitchen. Either way, that outer structure is gone before any meaningful thought is given to it. The pod wall of Pisum sativum is exceptionally rich in soluble fiber, particularly pectin, which forms a gel-like matrix in the digestive tract. That gel physically surrounds food particles and slows the enzymatic breakdown of starch. The result is a flatter, more gradual rise in blood glucose after a meal, but there is more happening than just fiber. Pea pods also contain significant concentrations of polyphenols, including catechins, epicatechins, and procyanidins, which inhibit alpha amylase and alpha glucosidase simultaneously. When both enzymes are partially inhibited at the same time, the reduction in post-meal glucose elevation is more pronounced than when either is targeted alone. In traditional European and Asian cooking, pea pods were not wasted. They were simmered into broths, dried into teas, added to soups whole. The pod was considered part of the food, not packaging around the food. That distinction between pod as food versus pod as waste is a modern invention. And like many modern inventions in food culture, it may have cost us something we did not realize we were losing. Number 8 is the acacia pod, and this is where the story starts moving into territory that genuinely surprises people. There are hundreds of acacia species distributed across Africa, Australia, the Middle East, and South America. Many produce pods that have been used as food and medicine across those regions for thousands of years. What drew modern researchers to acacia pods initially was their fiber content and prebiotic effects. That research is solid and well established. But the more interesting discovery came when researchers examined what happens to glucose metabolism with regular acacia pod consumption. Acacia pods contain polysaccharides that modulate the gut microbiome in ways that produce short-chain fatty acids, particularly butyrate and propionate. These fatty acids then interact with intestinal cells to stimulate production of hormones like GLP-1 and PYY, which signal the pancreas to regulate insulin secretion more efficiently. This is a completely different pathway than what we saw with the bean or pea pod. Instead of directly slowing glucose absorption, acacia pods appear to influence the hormonal environment that governs how the pancreas responds to glucose in the first place. The mechanism is indirect. The downstream effect on glucose control is real and measurable. If you are finding this useful, subscribe to the channel and drop a comment below telling me which pod surprised you most so far. We go this deep on topics like this regularly, and the next few items on this list go even further. Number 7 is the African locust bean pod, and this one carries a history that reaches further back than most people expect. The African locust bean tree, Parkia biglobosa, grows across the Sahel region of West Africa from Senegal to Sudan. For centuries, the pods and seeds have been central to the food systems of communities across that region. The fermented seeds produce a condiment called dawa dawa or iru, still widely used today, but it is the pod itself that contains something researchers began examining more carefully in the late 20th century. The pod pulp of Parkia biglobosa is sweet, historically eaten directly or dissolved in water. Detailed analyses found it rich in tannins and phenolic acids, including gallic acid and ellagic acid, that inhibit alpha glucosidase enzymes with notably higher potency than many of the compounds we have already covered. The gallic acid content has been examined in multiple cell and animal models for its ability to reduce glucose absorption and improve insulin sensitivity through pathways involving the GLUT4 glucose transporter, the primary channel through which cells take up glucose from the bloodstream. What makes this finding significant is the concentration. The pod appears to contain these compounds at levels that produce measurable metabolic effects in animal models, not trace amounts requiring massive consumption. The traditional communities that used this pod as food were, whether they knew the biochemistry or not, consuming meaningful quantities of compounds with genuine metabolic activity. That pattern keeps emerging across this list. Traditional use was not random, it was observational. Over generations, communities noticed which plants and which parts of which plants produced effects that mattered. They did not have the vocabulary of flavonoids or GLUT4 transporters, but they had thousands of years of watching what happened when people ate certain things in certain ways. The scientific vocabulary arrived recently. The knowledge itself is ancient. Number 6 is the tamarind pod. Tamarind is everywhere. It is in Worcestershire sauce, in pad thai, in agua fresca across Mexico, in chutneys across South Asia. Most people who consume tamarind consume only the pulp, the sticky sour sweet paste extracted from inside. The hard, brittle pod shell gets cracked open and discarded, and that shell contains something the pulp does not. The outer shell of Tamariндus indica contains a profile of polyphenols including procyanidins, luteolin, and apigenin studied for anti-inflammatory and antioxidant properties. In diabetic animal models, tamarind pod components produce reductions in fasting blood glucose, improved glucose tolerance, and reduced markers of oxidative stress in pancreatic tissue. The mechanism appears to involve both enzyme inhibition and direct protection of pancreatic beta cells, the cells responsible for producing insulin from oxidative damage. That last part is worth pausing on. Most mechanisms we have covered so far operate downstream of insulin production. They slow glucose absorption or improve insulin sensitivity. But protecting the beta cells themselves, the source of insulin production, is a different kind of action. It suggests the tamarind pod may be working at a more fundamental level in the metabolic system. We are at the midpoint of this list. The next 6 pods are where the research becomes genuinely remarkable. The mechanisms grow more complex, the evidence base gets larger, and the final entry has accumulated decades of controlled human research. We are not close to finished yet. There is a plant that grows in semi-arid regions across India, Pakistan, and parts of the Middle East that most people in the Western world have never heard of. It produces long, slender pods. Those pods are harvested, dried, and ground into a fine powder used as a food thickener, a stabilizing agent in ice cream, a texture modifier in processed foods, and a component in industrial applications from paper manufacturing to textiles. It is called gyar, and its pods contain something that has generated more nutritional research than almost any other leguminous structure in this category. Guar pods from Siamopsis tetragonaloba are the source of guar gum, extracted from the seed endosperm, but the whole pod contains a type of soluble fiber called galactomannan at concentrations among the highest found in any plant structure studied for metabolic effects. When galactomannan contacts water, it forms an extraordinarily viscous gel, more viscous than almost any other dietary fiber. That gel slows gastric emptying, reduces the surface area available for glucose absorption, and inhibits the enzymes that break down carbohydrates from accessing their substrates. The cumulative effect is a significant reduction in post-meal glucose and insulin responses, not marginal. Clinical trials using guar supplementation in people with type 2 diabetes have shown reductions in fasting blood glucose, improvements in HbA1c over 12-week periods, and reductions in LDL cholesterol. Multiple trials, consistent results. The traditional use of guar in South Asian cooking predates all of this research by centuries. Guar beans were a staple crop in Rajasthan, consumed whole, pods included, by communities where discarding any part of a plant was an unaffordable luxury. The knowledge that this plant had beneficial effects on digestion was embedded in traditional practice long before anyone had a laboratory to confirm it. What modern science added was precision, the ability to identify which compounds were responsible and what the dose-response relationship looked like. Number 4 is the carob pod, and here the history becomes almost disorienting in its depth. Carob, Ceratonia siliqua, has been cultivated in the Mediterranean basin for at least 4,000 years. Ancient Egyptians used it. Greek and Roman texts reference it. It appears in biblical accounts as the food that sustained John the Baptist in the wilderness, which is why carob pods are sometimes called St. John's bread in traditional European contexts. The word carat, as in the unit used to measure gold and gemstones, derives from the Arabic word for carob seed. Because the seeds were so uniform in weight, they were used as a standard unit of measurement in ancient jewelry trading. 4,000 years of human relationship with this plant, and the pod, the sweet, chocolatey tasting outer shell, was eaten directly, ground into powder, and prepared in dozens of traditional ways across the Mediterranean world. Researchers found that the carob pod contains an unusually rich combination of insoluble and soluble fiber alongside condensed tannins, primarily procyanidins. The ratio of fiber types produces synergistic effects on glucose metabolism that neither fiber type produces as strongly in isolation. Clinical studies examining carob pod fiber supplementation found reductions in post-meal glucose peaks of 15 to 20% compared to control groups. That is a clinically meaningful reduction produced by consuming the ground outer shell of a fruit that ancient civilizations ate as a snack. But the mechanism that makes carob pods scientifically unusual goes beyond fiber and tannins. The pods also contain a compound called D-pinitol, a cyclitol with insulin mimetic properties. Insulin mimetic means it appears to activate some of the same cellular pathways that insulin activates, specifically the signaling cascade that promotes cellular glucose uptake. In animal models, D-pinitol reduced blood glucose through mechanisms that operate even when insulin secretion is impaired. This is a fundamentally different category of action. The other pods on this list influence how glucose enters the bloodstream or how the body signals a response to glucose. D-pinitol appears to directly influence how cells take up glucose, mimicking one of insulin's primary functions at the cellular level. That is a specific molecular interaction documented in peer-reviewed literature. And yet carob is known almost exclusively as a chocolate substitute. There is a larger pattern worth naming directly. The parts of plants that end up in waste streams tend to be the parts with the highest concentrations of protective compounds. From an evolutionary biology perspective, the structures that protect seeds need to deter insects, resist microbial invasion, and survive weather extremes. The chemistry that evolved to do all of that turns out to overlap significantly with the chemistry that influences how mammalian metabolic systems process glucose. We were not the intended beneficiaries, but we can be. Number 3 is the moringa seed pod, and at this point the science moves into territory that even people who follow nutritional research closely tend not to know well. Moringa oleifera, sometimes called the drumstick tree, is native to the Indian subcontinent and cultivated across tropical and subtropical regions of Africa, Asia, and Latin America. The leaves have received significant nutritional attention. The seeds have been studied for water purification. But the pods, the long-ridged seed cases that give the tree its drumstick nickname, have a research profile that is separate from both and in some respects more interesting. Moringa pods contain isothiocyanates, specifically moringin, along with quercetin glycosides and chlorogenic acid derivatives. The isothiocyanates have been studied for their ability to activate NRF2, a transcription factor that regulates the expression of antioxidant and anti-inflammatory genes. When NRF2 is activated, it reduces oxidative stress in insulin-sensitive tissues including skeletal muscle, the liver, and adipose tissue. Why does that matter for glucose control? Because oxidative stress and chronic inflammation are now understood as primary drivers of insulin resistance. They damage the insulin receptor signaling pathway at the cellular level, impairing the ability of cells to respond normally to insulin even when insulin is present in adequate amounts. By reducing oxidative stress in those tissues, moringa pod compounds appear to help restore normal insulin sensitivity by addressing the root mechanism of impairment rather than compensating for its downstream effects. What is remarkable about moringa pods is not one mechanism. It is several operating simultaneously. Glucose absorption, cellular insulin sensitivity, oxidative stress, and inflammation through distinct but complementary pathways. Each pathway would be interesting alone. Together, they create an overlapping protective effect that is difficult to achieve with any single pharmaceutical compound because each drug typically targets only one pathway at a time. We are almost at the top of this list. 2 pods remain. The evidence base for what is coming is substantially larger and more rigorously tested in human subjects than anything we have covered so far. The next pod has a name most people recognize. But what most people know about it is only a fraction of what makes it scientifically remarkable. And the pod itself, the actual outer structure, is almost never the part that gets discussed. That is about to change. The mesquite tree does not look like much. Scrubby, thorny, low to the ground in the way that plants in harsh environments tend to be. It grows across the southwestern United States, Mexico, and large parts of South America. Ranchers in Texas have spent enormous effort trying to eradicate it from their land. For thousands of years before any of that, indigenous peoples across the American Southwest built entire food systems around it. The mesquite pod is long, yellowish tan, slightly curved, with a sweet starchy pulp surrounding the seeds. The Tohono O'odham, the Pima, the Apache, and dozens of other nations harvested mesquite pods as a staple food. grinding them into flour that could be stored through winter. For some communities in the Sonoran Desert, mesquite pods were a primary caloric source for months of the year. Mesquite pod flour has a glycemic index of approximately 25. White bread sits around 75. Glucose itself is set at 100. That means mesquite produces a post-meal blood glucose rise roughly 1/3 of what white bread would produce. The mechanism involves exceptionally high soluble fiber content. Fructooligosaccharides fermented slowly by gut bacteria rather than digested for immediate glucose, a protein content of 11 to 17% that slows gastric emptying independently of fiber, and compounds that in animal studies appear to support beta cell recovery and improved insulin output after experimental damage. When traditional foodways were displaced by commodity foods, the pods stayed behind on trees that people now consider weeds. The nutritional system that sustained communities for thousands of years became agricultural debris. And that brings us to number 1, fenugreek. The seeds appear in South Asian, Persian, North African, and Ethiopian cuisines. Trigonella phoinum graecum has been cultivated for at least 6,000 years, appearing in ancient Egyptian medical texts, in Greek and Roman agricultural records, in Ayurvedic and Unani medicine. Almost all of that documented use focuses on the seeds. The pod, the outer shell, is almost universally discarded. It never makes it into spice jars or supplement capsules, and it contains a concentration of the very compounds that make fenugreek metabolically remarkable, in some analyses at higher levels than the seeds themselves. The fenugreek pod contains 4-hydroxyisoleucine, an unusual amino acid found in significant concentrations almost nowhere else in the plant kingdom, studied in over 40 controlled experiments. It directly stimulates insulin secretion from pancreatic beta cells in a glucose-dependent manner, meaning primarily when blood glucose is elevated, reducing the hypoglycemia risk associated with non-selective insulin secretagogues. The pod also contains galactomannan fiber at concentrations comparable to GOR, slowing glucose absorption while the amino acid simultaneously amplifies the insulin response. Two mechanisms working together. One slows glucose in. The other sharpens the body's response to whatever does enter. On top of these two, fenugreek pods contain steroidal saponins including diosgenin and yamagenin, studied for their ability to improve cellular insulin sensitivity through the PI3K and AMPK signaling cascades, the same pathways targeted by some of the most widely prescribed diabetes medications. A meta-analysis examining 14 randomized controlled trials on fenugreek preparations found statistically significant effects across all major glycemic markers. 14 trials, multiple countries, consistent results. And the pod, which may contain higher concentrations of the primary active compounds than the seeds, is the part that ends up in agricultural waste every single harvest. 6,000 years of documented human use. And the outer casing of its seed pods has barely been examined as a distinct entity. Think about what this list actually represents. Not exotic plants from remote corners of the world. The opposite. Bean pods, pea pods, tamarind, carob, fenugreek, plants that appear in grocery stores, farmers markets, and traditional kitchens across dozens of cultures. Not rare, not obscure. Ordinary enough that most people have handled them, used the part they recognize as useful, and discarded the rest without a second thought. The compounds we covered throughout this list did not emerge from pharmaceutical laboratories. They evolved over millions of years as part of the chemical architecture of plants that have lived alongside human beings since before agriculture existed. When traditional cultures consumed these pods in broths, teas, and whole food preparations, they were not performing unverified folk medicine. They were applying accumulated observational knowledge about how specific plant structures affected how people felt and how their bodies responded to food. What changed was not the plants. The plants are still there, still producing the same compounds they always have. What changed was the context. Industrial food processing created a world where the edible seed became the product and everything surrounding it became waste. The metabolic consequences of that shift are still being measured, but they are measurable. None of this means that eating pods resolves a metabolic condition that has developed over years or decades. What it means is that plant chemistry we have been systematically removing from our food supply had functions that we are only now beginning to fully document. And some of those functions were directly relevant to the metabolic challenges that are now among the most prevalent health concerns in the industrialized world. You can walk through a market now and see these plants differently. The tamarind pods in a bin, the fenugreek bundles, the carob pods on a specialty shelf, the fresh bean pods at a farmer's market stall. They are the same objects they have always been. But what you know about them is different now. If this video changed how you think about the plants you handle every day, subscribe and hit the like button. It helps this kind of content reach more people who are looking for exactly this. And leave a comment telling me which pod on this list you had never heard of before today. I read every single one.